Biotechnologies enable clean, environmentally friendly manufacturing processes. Microbes can be used to produce industrial products and materials, whilst generating less waste and greenhouse gases. One family of microorganisms, Magnetotactic bacteria, align themselves along magnetic poles using tiny iron-rich organelles called magnetosomes. This unique property can be used in self-replicating nanomagnets to replace traditional chemical magnetic nanoparticles or develop entirely new technologies. Applications include cancer treatment, drug delivery, energy generation and metal capturing. However, large-scale production of sustainable, next-generation biological nanomagnets remains challenging. Researchers must address the growth requirements of the bacteria and maximize the accumulation of magnetosomes within each cell. Magnetosome formation is not fully understood and therefore difficult to optimize. Dr. Alfred Fernando Castaigne of Aston University is investigating new cultivation techniques characterizing magnetosome formation and determining how iron is incorporated into the cell. Preliminary research has revealed variation in metabolism and iron incorporation, enabling the fine-tuning necessary for lab-scale production, which can be applied to future industrial manufacture. Dr. Castaigne plans to make his findings publicly available, unlocking the potential of magnetotactic bacteria and their magnetosomes as sustainable technologies for next-generation biomedical and biotechnology applications.